Hi everyone, this is Miss Lasar, and today I'm going to be teaching you about osmosis and diffusion. First, some reminders about your notes. You're taking handwritten notes during this lecture video, and they'll be in outline format again. So I've pre-formatted all of the notes so that your notes will look something like this on your paper when you're done. Some tips: make sure your numbers and letters are vertically aligned. Pay attention to how much you've tabbed in and keep it consistent. If it's black text, you should write it down. And if it's blue text, it's optional, maybe a tip, maybe a fun fact. You can choose whether to write that down or not. Finally, I might go too fast. I'm probably going to go too fast. That's okay. Pause the video to write down notes when you need to. The most important thing is that you have all the detail that's in the video in your notebook. Let's get started. So, Section A. Important vocabulary. Here's our first important vocabulary term. Number one, a molecule. You probably are familiar with this one. A molecule is a substance made of multiple atoms bonded together. We can have really small molecules made of just 3 atoms, like water, made of oxygen, hydrogen, and hydrogen. Or we can have somewhat larger molecules like caffeine that has many more atoms bonded together. What's important to remember here is that everything you see around you, including you and the bodies of everyone you know, all things in the universe are made of molecules. So when we look at a glass of water, what we're really looking at is billions and billions and billions of tiny microscopic water molecules that are all together in one big clump. When I look at you, you are made of many, 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 many molecules that are way too small for us to see, and they make up you and everything about your body. So that's just important to remember because everything we talk about today and in the future of biology class is going to be made of molecules. Important vocabulary number 2, a solution. A solution is a mixture of 2 or more substances where a solute is dissolved in a solvent. Ooh, those are 2 new words as well. Number 3, a solvent. A solvent is the primary liquid in the solution. And number 4, solute. This is the substance dissolved in a solution. We've got an example of a solution, solvent, and solute on the screen. I'll talk you through it, and then I'll talk you through a couple more examples. Here, our solution is going to be salt water. To make that salt water, we mix our solute, salt, with our solvent, water, and together those are salt water. Let's look at some more solutions. Here's another solution, one of my favorites. This is coffee. Okay, just by looking at this, what is the solvent? It is water. Tip: the solvent is almost always going to be water. In biology, it will always be water. Once you get into chemistry, you might see some different solvents like ethanol or benzene. What is the solute in this situation? Well, what's dissolved in the water? Coffee molecules. The coffee itself is dissolved in the water to make our solution of coffee. But what if it's sweetened? Then we have another solute. It would also be sugar. So solutions will have one solvent only, but they can have multiple solutes. Many, many solutes are in biological solutions like blood. Here's another example: seltzer water. Seltzer water is a solution which might be surprising. What's dissolved in here? Well, let's work backwards. What's the solvent? As usual, the solvent is water. And what's dissolved in it? It's carbon dioxide, which is a gas. Carbon dioxide is what makes seltzer and carbonated beverages fizzy. And this is an interesting thing to think about. You can have a solute that is a gas, not a solid like sugar or coffee grounds. Now we're ready to move on to number 5, concentration. The number of molecules in an area or volume. Let me show you 2 examples. Here is an area of low concentration. Each circle represents a molecule, and they're pretty spread out. There's few molecules in this area. This is an area of high concentration. There's far more molecules packed into this area. When I think of concentration, I think about how packed together the molecules are. Are they very packed together, highly concentrated, or are they more spread apart, less concentrated? Here are 2 real examples. Human blood is a solution with a concentration of 0.9% salt. What does that mean? Well, 0.9% of the solution is salt. That's the solute. 99.1% is other stuff. Most of the other stuff is water, but there's also sugars, proteins, cells, and other stuff in your blood. Here's a slightly simpler solution: salt water. Ocean water is a solution of many different salts dissolved in water. Ocean water has a solute concentration of 3,500 milligrams per liter. That means that in 1 liter of water, there is 3,500 milligrams of dissolved salts. So, those are some different ways that you might use the word concentration, solution, and solute in sentences and real examples. Term 6, a semi-permeable membrane. This is a boundary that only certain molecules can pass through. And there's one really common example of a semi-permeable membrane that we're going to be referring to for the rest of the lesson, and that is the cell membrane. All cells, all of your cells, all plant cells, all bacteria cells, all fungi cells have a semi-permeable membrane around them that regulates what enters and leaves the cell. Here's what that kind of looks like. So you can see in yellow, this is our semi-permeable membrane. The red things aren't moving. Those are molecules that can't cross the semi-permeable membrane. It's a barrier to them. But you can see these blue molecules, they are able to cross the semi-permeable membrane. So for some reason, and you'll learn about why in chemistry, some molecules can cross the semi-permeable cell membrane and some can't. Let me show you another example that isn't the cell membrane, but is a semi-permeable membrane that's used in medicine. It's called dialysis. So this is blood. Blood, we already said, was a solution. There's a bunch of stuff in it. For example, water in blue, red blood cells, white blood cells, and toxic chemicals. And in a healthy person, your kidneys will filter out all of the toxic chemicals and all the extra water that's in your blood on a normal daily basis. But if you have kidney failure, your kidneys can't filter those things, the extra water and the toxic chemicals, out anymore. And so you have to go on dialysis. What happens in dialysis is your blood is slowly removed from your body and filtered through dialysis tubing before it's put back in your body. Here's dialysis tubing. You can see that it's no longer solid lines. The dialysis tubing is actually a semi-permeable membrane. And if you look at the size of the holes in the semi-permeable membrane, what could fit through? Well, the water is small enough to fit through. The toxic chemicals are small enough to fit through. But the red blood cells and the white blood cells, which are good things to have in your blood, those are too big. Those won't fit through. So, as your blood is run through dialysis tubing, extra water and toxic chemicals will go through the semi-permeable membrane, leaving all this good stuff left in the blood, and this cleaned blood will be returned to the person's body. And that's how dialysis works. Pretty interesting and important example of a semi-permeable membrane. All right, now we're ready to talk about our second topic, diffusion. So now we're on capital letter B. Make sure it's vertically aligned with the capital letter A. Diffusion. This is a vocabulary term, so like the others, underline it, make it stand out. Diffusion, number 1, is the net movement of molecules from an area of high concentration to an area of lower concentration. Here's what it looks like in picture form. If we start off with our molecules highly concentrated, really packed together, on their own, without us doing anything, they will spread themselves out to where it was less concentrated. At the beginning, all this surrounding space is much less concentrated. There's none of these red molecules there. They'll spread themselves out until they're evenly spread out through the space. They do this by just moving around randomly, because molecules are always moving around randomly, even if you don't see it or if it doesn't feel like it. So here you can see our molecules started off all packed together, all clumped together. And now they're moving around randomly. And just by moving around randomly, they're spreading themselves evenly through the rest of the space. There you go. You can see it again. This is the process of diffusion. And you've definitely seen this in action. You experience this all the time. Let me give you some examples. Still in diffusion. Now, number 2, examples. First example little a, food coloring diffusing into water. If you put a drop of food coloring in a glass of water without stirring it, without touching it, without moving the glass, that food coloring will spread itself out until the whole cup of water is evenly colored by that food coloring, and there's no more clumps or regions that are darker than the others. That's diffusion of the food coloring through the water. Here's another example, smoke diffusing away from a fire. You know that when there's a fire happening in one location, the smoke doesn't stay right on top of that fire. Even though it's produced by that fire, it moves of its own accord without wind blowing it, without any energy being added to it. The smoke will diffuse through the air around the fire until you, standing 100 feet away, can smell the smoke from the fire. Similarly, if you spray air freshener in a small corner of a room, if you wait for a little bit, that air freshener will spread itself out. You'll be able to smell it throughout the whole room. Even without mixing the air or pushing the air freshener, those molecules will move of their own accord. So now we're ready to talk about a very specific example of diffusion called osmosis, capital letter C, osmosis. This is the main topic for the rest of the video. Make sure you underline this. This is the most important word from today. Osmosis is the diffusion of water through a semi-permeable membrane. So now you're seeing why we started with concentrations and permeable membranes and diffusion. It's all going to come together here on osmosis. Let me show you what I mean. This is a visualization of osmosis. The blue molecules are water molecules, and you can see them diffusing from where they're highly concentrated to where they're less concentrated. Here's another way that I like to think about osmosis. This is an alternative definition that you're welcome to write down. It might be helpful. The movement of water molecules from higher concentration on one side of the membrane to a lower concentration on the other side of the membrane. And they're going to spread themselves out until they're equally distributed on both sides. Let's look at this again. We've got two cells that are inside a solution, some sort of solution. We don't really know what's in the solution or what's inside each of the cells, besides the percentages of water and the percentages of solutes in each location. Here's a tip. Osmosis is all about water moving. Water will move to the area with the higher solute concentration, which is also the area with the lower water concentration. It's the same thing. Higher solute concentration will also be where there's a lower water concentration. So if we look here, this cell has a 10% solute concentration versus the solution, which has a 5% solute concentration. Okay, so water will move to the area with the higher solute concentration. That means that water is going to flow into this cell. Think about what's going to happen to this cell size. Here, we've got a 2% solute concentration compared to 5%. So 5% is the bigger solute concentration here. Water will flow out of the cell to where there's a higher solute concentration. Think about what that will do to the cell. So here are some examples of osmosis in your life. Section 2, examples. Example A. Water flows from your large intestine into your cells when you drink water. So your large intestine is a tube, and what you're seeing here is a cross section of that tube. I've taken it and I've cut it in half so you can see the layer of cells on the top, the layer of cells on the bottom, and then all the stuff that's inside your large intestine and moving through it. Let me tell you about it. The water concentration inside your cells is 92%. The water concentration inside the small intestine after you've drunk a bunch of water is 98%. Ooh, higher water concentration here. So water will move to where it's less concentrated. That means that water is going to move from the inside of the small intestine into your cells. That's a good thing. That's how you get water from the water you drink into your body. It diffuses through osmosis from your small intestine into your body cells. But the reverse can also happen. Example B, water flows out of your cells into your large intestine when there's too much lactose in your large intestine. Okay, so lactose is in things like milk and cheese and dairy products. And if you're lactose intolerant, you can't break it down. And that means that you're going to eat those things with lactose. The lactose won't get broken down. It won't get used for energy. It'll just stay until it's in your large intestine. And then we have a situation like this, where there's all this lactose in your large intestine, along with water and other stuff. Now, the water concentration inside our cells is 92%, but the water concentration inside the small intestine has dropped to 89% because there's so much other stuff, so much lactose in there too. Now, water will move to where it's less concentrated, which is now inside the small intestine. This is a lower concentration than what's inside the cells. And so water is going to move from the cells into the small intestine. That means that, large intestine. And that means that you're going to have water rushing into your intestines and you're going to have diarrhea. Tough. Now we're ready for section 3, some more osmosis vocabulary. Here's the first vocabulary term: a hypertonic solution. A solution with a higher solute concentration than the one it is being compared to. So I want you to look at this cell in this solution. This cell is in a hypertonic solution. I know that because the solution has a higher solute concentration than inside the cell. So using these terms correctly in a sentence is really important. That's why I'm going to show you a couple examples of how to use each of these terms. Here's the first one. I would say this cell is in a hypertonic solution. I could also say the outside solution is hypertonic to the cell. This is a comparison. Or I could say the outside solution is more concentrated than the solution inside the cell. That gives the same meaning, even though I'm not using the term hypertonic. So what if the opposite is true? What if there's a lower solute concentration outside the cell? Well, then we're looking at lowercase b, hypotonic solution. A hypotonic solution is a solution with a lower solute concentration than the one it's being compared to. So this is the opposite of hypertonic. I would say in this situation, the cell is in a hypotonic solution. Look at the solute concentrations. The solute concentration on the outside is lower. So the cell is in a hypotonic solution. I could also say the outside solution is hypotonic to the cell. Or I could describe it. The outside solution is less concentrated than the solution inside the cell. There's one more situation. What if the solutions have equal concentrations? Well, then we're looking at lowercase c, an isotonic solution. An isotonic solution is a solution with an equal solute concentration to the one it's being compared to. Here we go, 23%, 23%. This cell is in an isotonic solution. The outside solution is isotonic to the cell. The outside solution has the same concentration as the inside of the cell. All of these sentences mean the same thing. One last vocabulary term in this section, lowercase d, equilibrium. Equilibrium is another way of describing a cell that's in an isotonic solution. Equilibrium is when two areas have equal solute concentrations. So here I would say the cell and outside solution are in equilibrium. And here's why equilibrium matters. Osmosis will happen until cells are in equilibrium with their surroundings. Let me show you what I mean. Now here we're not in equilibrium. The cell is in a hypotonic solution. I would say a cell in a hypotonic solution will go through osmosis, will in this case gain water until it's in equilibrium with its surroundings. How do I know it's going to gain water? Because water will move to where there's more solute, where there's a higher solute concentration. And so water will flow from the surroundings into the cell. And that's going to happen until the solute concentrations are equal, until we've got 2% solute on the outside and on the inside. We're going to have to add a lot of water to make those concentrations equal. So let's talk about that flow of water and section 4, how osmosis affects cells, how that flow of water affects cells. Detail A, animal cells need to be surrounded by an isotonic solution. Here's why. You might have already inferred what's going to happen when we put cells into isotonic, hypertonic, or hypotonic solutions. Here's an animal cell in an isotonic solution. That means that the concentration of solute inside and outside are the same. And so water is actually going to flow into the cell, but it's also going to flow out of the cell. Water molecules are always moving, they're always flowing. So they're going to flow into the cell and out of the cell at the same rate. And the cell is going to stay the same size. This is what we want. We want all the cells in your body to stay the same size because if we put them in a hypotonic solution, where the inside of the cell is more concentrated than the outside of the cell, there's more water outside the cell, there's less solute outside the cell. Well, in this case, the direction of water flow will be into the cell, from where there's more water to where there's less water, from where there's less solute to where there's more solute. And as that water flows in, think about a water balloon expanding. The cell is going to grow and grow and grow and swell and swell and swell until it bursts. And when a cell bursts, it dies. So if animal cells are in hypotonic solutions, they will swell until they burst and die. Ooh, this is a big issue. And there's also an issue with hypertonic solutions. Here's a cell in a hypertonic solution. There's more solute outside the cell than inside the cell. So water will flow out of the cell to where there's more solute. When the cell is losing water, what happens to it? Ooh, it shrinks, shrivels, and it dies. So an animal cell will die if it's in a hypotonic or hypertonic solution. Here's some real life examples of how we can use that. Saline used in hospitals, that's what you would have in an IV, contains 0.9% salt so that it is isotonic to human blood, so that it doesn't make blood cells swell or shrink. It keeps blood cells the size that they should be. But we can also use it to kill things like slugs. Putting salt on slugs creates a hypertonic environment around the slug. There's too much solute around the slug. This causes water to rush out through osmosis, killing the slug. Notice how I used the word osmosis and the word hypertonic in these sentences. Think about how you would be using these words in sentences. Consider writing this down as a model or a framework for the right way to use osmosis and hypertonic in sentences. What about plant cells? Well, last detail, plant cells need to be surrounded by a hypotonic solution. Ooh, interesting. Here's why. Here are the three possibilities for plant cells and what's going to happen to the plant based on the solution it's in. On the left, we've got a plant in a hypertonic environment. There's less water on the outside than there is on the inside of the cells. In that case, water is going to rush out of the cells, and the cells are going to get super, super floppy. They're going to shrivel up, and the plant will be super wilty, and it could even die. One thing I want you to notice is that there's this outer boundary of the plant cell, and even though the interior is shriveling up, the exterior is not. That's because plant cells have something around them called a cell wall. Different than the cell membrane, the semi-permeable cell membrane, that's actually this interior soft squishy thing. The cell wall is on the outside. It's very hard and firm, and it doesn't lose its shape very easily. Here is a plant cell in an isotonic environment. Here, water is moving equally into and out of the cell, and the plant cells will be expanded. They'll look normal, but they'll be kind of like animal cells. They'll be a little bit soft and squishy. When you think of plants, you don't think of soft and squishy. This soft and squishy plant is actually still going to be a little wilted. What plants need is a hypotonic environment where water is constantly rushing into the cells. Because plant cells, unlike animal cells, don't burst. That cell wall is so strong that the water pressure will expand the cell and then it'll just stay fully blown up. It'll be much firmer, it'll be much stronger, and it won't burst. This expanded, swelled plant cell is actually what gives plants their strength. Strong plants are plants in hypotonic environments. If they're in isotonic or hypertonic environments, they'll be a little wilted. That's the information that we have for you this week about osmosis and diffusion. I hope you enjoyed this lecture video. And if you missed anything, remember to go back, rewatch any sections that you need to so that you've got a full set of notes. See you in class.